Good evening, everyone. Thank you for joining. I hope that others will also join because we have an interesting talk ahead of us on an unusual, uncommon topic that many people might not know, might not have heard about. Dr. Marasinghe, Dr. Kalpani Marasinghe is a lecturer in the Colombo faculty, science faculty, and she's done a PhD on the Bryozoans, these rather beautiful but very small, insignificant animals that are totally marine. And those of you who have listened to the pre-lecture chat would have realized that very little work on this group has been done in Sri Lanka. There are lots of groups of animals that very little work has been done in this country. So let's get going. Kalpani, you can put your full screen on and start your talk.
Okay.
If anyone has any questions, please put them on the chat. And once she is finished her lecture, we'll have a discussion and have an opportunity to answer any question that you have. Go ahead, Kalpani.
Thank you very much, sir. Thank you very much for inviting me for this session. And it's like something that I'm really interested in as one of the creatures that I came across during my PhD period. And I first encountered this particular phyla during 2014 when I was started my PhD. So before that, I only know only knew two species out of this phyla. And then when I came across this sheet-like, the lichen-like encrusting sheet, I was wondering what this particular species is, what this particular species category is. So I just took a piece out of it and put under the light microscope, and then I observed, like, I was really like, it was like a very interesting moment for me. No, I never forget. And starting from that day, I was like addicted to this particular species, the morphology and all that. So I learned myself, and then I went through a lot of literature, and I studied a lot. Maybe, like, most of the time I was like with the microscope and studying about this, trying to identify what I found. So based on that findings, I'm presenting today's lecture on bryozoans. So I will start my session. Right. So if you're an like kind of a someone who is like fascinated about these underwater like creatures that we come across, these beautiful, colorful species, you might have come across any of these species. Some of them are freshwater, and most of them are like found in marine waters. So sometimes you could have misidentified these as corals. And let's see what these particular creatures are, and let's see how we can differentiate these from the other species, and what are the, like, there are so many interesting facts and interesting potential research area when it comes to this particular phyla. So this could be something that you have already came across, these beautiful, colorful creatures. But when you look at this slide, the next slide, you will see what made me interested about this particular species. So these are a couple of microscopic photographs that I took during my analysis. These are microscopic images. And these are a couple of species that I came across. When you're looking these specimen through the microscope, you find, like, teeny tiny little details that make it, like, more interesting. You are like, we want to identify all of these, and try to see what this particular species is. So this is something that made me interest to start to learn about this particular species. In the bottom images, in bottom, you can see a couple of photograph where you can see this particular species attached to debris. And then in this middle blue color one, this background one, it's a fishy vessel where you can find these bryozoans attached. And then in the right hand side, you can see a propeller of a vessel where these may attach. So these are some of the stuff that come across when it's come to like, you know, what make it interesting for me, this particular species, not just their, you know, taxonomy and all that, but some of the, you know, impacts and some of the, you know, functions that this particular phyla have, right? So when it's come to this particular phyla, as I told you before, this came across as one of the member of what I studied during my PhD program. So I mainly, I have a PhD in zoology where I study mainly about biofouling community, and these bryozoans are one of the members of this biofouling community. So if you see a fouling community, it have this microfouling that we can't see from our naked eye, and then we have this macrofoulers, and when it is macrofoulers, we can see them. And when it comes to these macrofoulers, we can find hard foulers and soft foulers. These bryozoans comes under hard foulers, where we see, like, you know, the typical barnacles and, you know, mussels, like, you know, oysters and tube worms. Likewise, bryozoans are under this category, hard macrofoulers. So I saw a couple of these encrusting sheets over my study plates. So, and then I started to look at what these particular colonies are. And then I started identifying them. And then during my PhD program, I studied about how they develop through the time, and then how these particular species interact with other, organisms in the fouling community, and whether there are any non-indigenous species introduced to our country waters as primary introducers. So we could find a couple of these bryozoans that are introducers. From my PhD program, I will let you know more details in coming slides. Okay. So that what I wanted to discuss in this lecture. It's a story beyond their beauty. There are so many applications and importance of this particular phyla other than their taxonomic importance. So when it comes to their history, and there are many of the important facts that make it interested for the people who are interested in history. So there's something for everyone when it comes to this particular phyla, if I say. So if you're interested in medicine stuff, then there is something important about this phyla, because there are medicinal importance. If you are interested in architecture, there is this colony structure. It it will make you interested. And if you are interested in archaeology, then there is something in this particular phyla. So let's have a look at these beautiful creatures. And I hope this less so spoken species category will be not anymore a less spoken one. So you can have kind of an idea about what this particular phyla is, and the different animals that we can see in this phyla. Okay, right. So this sometimes, we come across these species in different, articles and all that in different words. So these are also known as moss animals, lace corals, and polyzoa, and then sometimes they are called as enteropta, and they are also called as modular or clonal organisms, and also some of the time they are called as lipoporatans. So these are a couple of terms that you can come across when you're looking through different sources about this particular phyla, right? So when it comes to these bryozoans, they are kind of colonial invertebrates, and they are, as I told you before, the most common term is most animals, and there are around about 6,000 extant species, but there are more extinct species, maybe around 20,000. And it's diverse and widely spread in marine and freshwater environment, and there are basically three different classes at the moment. That is Phylactolemata. We can find them only in freshwater. And then Stenolemata, we can find them only in marine. And then this Gymnolemata, the only class that I came across during my studies. And they are mostly marine, but we can find a few of the members in freshwater environment as well. Okay, so as I told you before, they are comprised a major component of fouling community. So when you talk about hull fouling and all these, like, industrial aspect when it comes to fouling communities, these bryozoans play a key role. So everyone who's talk about these fouling organisms, then bryozoan, they all know about these bryozoans. So they have a long history dating back to the previously it was said during the time that when I was doing my PhD, it was a Ordovician period, but recently they found it's from the Cambrian era as well, but there are some confusions. Let's talk about that one as well in later slides. So, but the saddest thing is this knowledge on bryozoan fauna is still poor for some part of the world, including Sri Lanka. It's less spoken, basically because of the difficulty in taxonomy, and it takes long time to identify these species. You have to consider very small features, and sometimes these features overlaps, and sometimes we can't find them only through one particular microscope. Sometimes you have to prepare specimens and see through scanning electron microphotograph and all sort of different methods you have to use. So it takes so long time to identify one particular species time, trust me. So it is obvious that this particular species is less spoken, right? So this particular species is like mostly colonial. There are a couple of species that are solitary, and they are suspension feeders. Mainly they rely on phytoplankton and sometimes zooplanktons as well. And they usually grow on rocky substrate. When you go to any kind of intertidal area, or maybe if you find kind of a piece of debris along the coast, just take it to hand and observe, then you will see some kind of encrusters that look like a lichen. But if you look closely, you will find compartments, very tiny compartments. So it's probably going to be a bryozoan. Okay, so they can observe in many of these solid surfaces, and their colonies range in appearance from gelatinous blobs to bushy or tree-like forms. So they are not just encrusting form, there are tree-like ones and then gelatinous one. Sometimes this gelatinous one can be misidentified with other gelatinous species like tunicates, and most of the time they look like a plant. So one of the common bryozoan that look like a plant we come across is bugula species. You might have heard of that particular species before, right? So so when it comes to this species, as I told you before, we can find them mostly in marine, but some of the species are there who live in freshwater. And I try to whenever I go to a stream or freshwater resource, I always try to find any kind of this freshwater bryozoan. So I couldn't find any of these. So it's again open for research. And this particular species have wide salinity tolerances, and they can be found in huge depths, maybe up to 8,200 meters, but majority live in shallow water areas. But when it comes to like, lagoons and brackish water areas, we can't find much of the diversity, because when I go like, to these lagoons and brackish water area, we can't come across this much of the diversity. And they are very fast growing. That is why it become a nuisance if it is non-indigenous and have kind of a potential invasive characteristic. And they have a high reproductive potential. And if you put a surface, like kind of an artificial surface onto the water, marine water, in within couple of weeks, you can find these bryozoans. So they can settle. There are always these larval stages in the water, and they come and settle down in these surfaces. So they settle on wide range of substrata. That is why when it comes to bioinvasions, this species become like very important. There are over 100 globally known invasive species of this particular non-indigenous species of this particular species category. So we can, as they can find in different kind of substrata, as I told you before. If you see some kind of a surface on the coast, then pick it up and see this lichen-like shape. It could be probably a bryozoan. Okay. Right. So when it comes to classification, as I told you, I'm not going to deep into this scientific aspect, but just to give a kind of an idea, there are mainly three classes: Phylactolemata, that is the most primitive one, and there is only one order extant. And then class Stenolemata, it's a calcified one. And then Gymnolemata, it is the most diverse one, where we can find order Calostomata and Tenostomatida. So these are this is the basic classification of this particular phyla. Right. So when it comes to their phylogeny, they have a common ancestor that is common for like mollusk and the common known ones, and they have a kind of relation with this annelid mollusk as well. And their closely related ones are Brachiosaura and Entoprocta. You might have heard of these particular species, and this when it comes to these lepoporatans, and you might have encountered these poronids. They are like, easily misidentified with some of the tube worms. And then this brachiopod, they have this lopopore as a common feature. That is why they are under the same category. So there are some, different classifications and, phylogenetic gradation within these groups, and some differences. But anyway, all these categories, they have this lopopore as a common feature. Right. I will talk about that in coming slide. So when it comes to their classification, we can classify each of these species into different categories because they have kind of common features and different features. That is why they fall into different categories. So when it comes to this Phylactolemata, the freshwater bryozoans, they don't have any mineralized skeleton. So you mostly find them as a fleshy muscular glob. So if you find something like that attached to, if you come across any of these in freshwater environment, please let us know. And these have a kind of a cylindrical zooid. If you look through them through the microscope, then you will find this compartment that are cylindrical. I will show you a couple of these images. And then I told you they have this specific feature here. If you can see here in the upper area, that is their feeding and excretory organ, that is lopopore, which is common feature of this particular phyla. So they have this, when it come to this category, this particular class have horseshoe-shaped lopopore. That is different from other classes. Okay, so that is one of the main differences that we use when it comes to classify this particular species into different classes. So then we have Stenolemata and Gymnolemata. They have circular lopopore. So when you see kind of their feeding appendage, when they put it out, then it look like a circular one. But if it is a Phylactolemata, it's like a U-shaped one. So by looking at this in the bottom image, you can see these differences. So these are some of the interesting facts that we use for the identification. So here you can see some of the real, images that we can come across of this particular different species of these categories, classes. So Phylactolemata, the interesting thing about this particular class is, I told you these are like mainly sessile one, they can't move. But the interesting thing is this Phylactolemata is this particular genus Lepopodella. They can creep, they have a slow movement, and they can move on the surface very slowly. It's like creeping movement, and other species are like are sessile. So in this order Chelostomata, you can see this couple of images. That's 3 images that are from my experimental panels. And these are the species I came across. And then we have this order Titostomatida and then Stenolemata. So when it comes to Gymnolemata, we can find these 2 orders, but I came across only about these 2 particular order, right? So when it comes to this particular colonies, you might wondering, how do they origin? So they origin from a primary individual known as the ancestrula. So if there is a new colony, then it will start with this particular individual called ancestrula. So it will bud and then it will develop this colony. Okay, so this each unit, I told you there's like small compartment. We call this each unit as zooid. That is the original organism of that particular colony. That is the interesting thing. So when it comes to different classes, these zooid are different. So when it comes to Gymnolemata, they have many different zooid types that are specified for different functions. That is what make it more kind of an advanced one. So when it comes to this each organism, they have 2 major regions, that is zoozoan and polypid. So I'm not going to describe these in detail. So when it comes to zoozoum is that you will see these encrustations. That is the outer protective case. That is the case. And the inside is the living part. That is called as polypid. So there are 2 major sections of this particular organism. So the interesting thing about this particular colony is that when it comes to these colonies on the surface, you see this as lichens. I hope you all have seen these lichens on trees. So when these colonies merge, like overlap, where 2 colonies never fuse, but 2 branches of the same colony can fuse. So when we are doing the identification and when you are doing the colony count for particular studies, to study about their growth and, colony count and all that, we might can, misidentify their number of colonies as well. So these are some of the important facts. So here you can see detailed figures about this particular zooid. So this is the detailed diagram. So I'm not going to dive into detail about this. So here you can see the living part and then the structure. Here you can see this compartment. I told you if you come across any of these surfaces, you will see this kind of compartments. And if you are lucky enough, and if it is a fresh colony, if it is alive, then put it into, if you see like there is like no damages to the colony and it's not weathered, then dip in the water and see whether there are any live one. And if you do underwater exploration, try to see, sometimes you can see through the eyes, if you closely look at. So you will see this beautiful flower-like thing coming out, so that they can feed. And then when they are inside, this is how you see it. So this is what really look like the colony inside of it. So when it comes to this, when as they are colony forming organisms, function of a colony depends on, different, they have kind of different work. So sometimes they have to feed, sometimes they have to have kind of a way to protect themselves, and they have to have a reproductive strategies to reproduce, and likewise. So each zooid can't do all these. So what they have, like the what they have done is they have different zooid specialized for each of these functions. So when it comes to a colony, most of the zooids are autozooid. Those are feeding zooids. And there are some other zooid called heterozooids. They are non-feeding one. And then there are particular zooid, that tiny like little organisms who are there for their protection. They deter small predators. And then when these colony get all that dirt on it, there are certain zooids that clean it. And then there are some other zooid called kinozooid. They fill up the space, free space. So they are compacted, but there are some spaces. So these kinozooids will fill up those, spaces and make it like a kind of a rigid structure. So that is what make it interesting. So when you see a piece of this colony, this is how the whole colony gonna look like. And when you look through the microscope, this is how you're going to see different organisms. And then the most interesting thing is when you look at these to the microscope, you get that is you actually have to see this under the microscope. You will see many different types of zooids. That is very interesting. I have seen all of these, but the bottom one is like very rare. I could only come across this only one time. But other than that, I could find all of these different kind of zooid in my samples. So I will quickly go through these features that make it more interesting. So this Lopopro, as I told you, they are suspension feeders. So this Lopopro is what they are used for their feeding. And they also use it for, reproduction as well. Putting away all their like grasping sperms during the fertilization and all that. They use that Lopopros as well. And so when it comes to the identification, when you look into a piece of this colony, there are like certain features that you need to focus on. So basically, we look at the colony color, if it is a fresh one. And sometimes we look at whether this colony are a single layer or whether there are multiple layers, likewise, because some colonies, they grow in different ways. So we look at colony shape and layers and whether there are any different kind of autozooid. From species to species, this autozooid and the different types of zooid I previously mentioned, all those are different. And then we see this operculum. It's something like something that they use to close when they are not put that lopopo, the feeding appendages out, they keep it this whole thing closed. That is what they use this operculum. And then we think about how they this zooid shape look like. Whether it's like a rectangle, like kind of a rectangular shape, or like kind of a oval shape, different type of shapes are there when it comes to these zooids. And whether there are any kind of spines, as you can see here in these yellow color arrows, whether there are any spines, and whether there are any avicularia, like different kind of zooids that use for defense. There are huge diversity in these avicularia as well. So I just want you to see these features to understand why it's so difficult to study about this particular phyla. But what makes it so unique about this phyla? But if you come to the right track of identify these different features, you just have to thoroughly go through these features and get familiar, and then you can easily identify these. Okay. So these are a couple of photographs that I took during my program. This is a Watercypora species. We could only encounter these colonies during 3 times. That's a globally well-known invasive species, but we could only find a couple of species. That is, in a way, a good sign, because we don't have that much of invasion in terms of this particular colonies. So it is like a very good sign, because in other countries, this has become like kind of a huge nuisance. So it is a good sign that we don't have much of density. So it's just couple of, it's just an non-indigenous species, not an invasive one to our country at the moment, right? So there are, I talked about this spine. So this particular species, that is Electra bengalensis, indigenous one. And we also called it as Arbopecula bengalensis. So this one is like, when I was first looking at this species, I was like, oh my goodness. Like, it's like a really, when you have to really see this one, it's like very complex one, and same time it was so beautiful with this complexity, and it's like these rib structures and horn like structures, like each of these features are actually identification features, whether these are branched or not, the length of these rib like spines, all these matters when it comes to the identification. So it's like huge lot of information, but that's what make it more unique and make it like very significant, right? And see, these are some of the features that I use for this identification, these spines. And then there are many different spines here and there. We have to see in which place they are located and compare those with other species of the same genus to identify which species is this particular specimen is. And then this is another species I encountered, and these are, as you can see here, they have a different colony zooid shape, and they have like kind of a sheet area that is like, kind of a membranous compared to other species. You can see the colony, the zooids are different from one another when it comes to these different genuses. That is what make it like, different. So that is what we have to focus when we identify this species. So here you can see these are some of the features that we need to consider, these small spines, and these we call these as denticles, and those are some of the things that we need to consider. And then there is this another significant feature in this bryozoan colonies, that is oviscel. These are brood chambers. When you see this, that means kind of we identify this as female zooids. So this is where they breed their larval stages. So when we come across these oviscel, that is again different from one genus to another. So there are certain, so if you look at this particular species, I actually wanted to focus on avicularia, but if you see these lines here, this kind of a circle area, that is again oviscels, but these are not significant as this particular species. So these are features are like, are very different. So when it comes to this avicularia, I told you they use this avicularia for defense. And these are like in different shapes, different sizes, and different positions. Even this avicularia, they have this mandible, the shape and the positioning of this mandible matters when it comes to identification. So it's like kind of complex, but it's like that what make them this particular different type of species unique, right? So these are some of the species I encountered, Cyanoplastra, and if you like, when you get familiar with this colony shape and the morphology and the zooid, when you look at this colony, you can identify it. So you get that level of uh, ability to identify this particular species, at least to the genus level, when you know a little bit about these zooid, and that is their different, this zooid characteristic make it different, like when you're looking at them in the naked eye. So if you can see this Cyanoplastra and Electra bengalensis, so if you can remember those rib-like structures, so this colony is kind of a little bit brittle. And then when it come to this Hippoporina indica, again, they have kind of a lumpy like zooid. So it kind of make it easier for us to identify. But this Hippoporina indica, sometimes you can get misidentified with this Schizoporella errata early colony. So, but when it comes to colony color, this particular species is like we can easily identify. And then this particular species, Celoporaria volcella, we can identify them easily because they have kind of a very rigid and needle-like appearance. So it's very easy to identify this colony as well. And then this Watercipora subtocota. And whenever I encounter any of debris or any of piece of hard substrate, I always look into this particular colony because I just want to look, whether they are whether there are any secondary introductions other than the primary introduction. So there can be a threat if we get more density of these colonies. And here you can see some of their SCM images that I took and that used for their identification. So when it comes to these colonies, these species, this Chrysoporella errata and Watercipora subtocata are two major species that are globally known invasive species. They are very nuisance. But luckily, this Watercipora subtocata, as I told you before, it's not that nuisance. But in the port, we observe this Schizoporella errata, a density that is could be a threat. I will show a couple of those images. So here, in the left-hand side, you can see Electra bengalensis, that is an indigenous one. In the right-hand side, you can see Schizoporella errata, it is a non-indigenous one. Trust me, this particular Schizoporella, it can cover up the whole experimental planet, 100%. And when the succession happened for a couple of years, I put my structures for a couple of years and to study the community succession. And sometimes this whole community wash off because of this particular Bryozoan species. So that is why I just want to emphasize the importance of studying this particular phyla. And there is kind of unforeseen threat if this kind of non-indigenous species come to our waters. It can threat indigenous species like Electra bengalensis. So when this is not from the same location, in the location where we can find this Chrysoporella errata with a huge density, we can't find that much of bengalensis colonies. You know, these colonies like a very soft one. It's very brittle, but this Chrysoporella errata is very kind of hard. So there could be a kind of threat in terms of their spread, right? And it is not enough if I don't show you any kind of a life cycle that they have. So they have this larval stage as well. So the special thing about studying their larval stage, I also studied their larval stages because when it comes to a biofouling community, not all species settle on a surface. Some species come and see whether this particular surface is good enough for me to settle or not. So if it is not good enough, they don't settle. They just go away. So it is important to study about these larval one to see whether there are any kind of a favoration for them to settle in particular substrate or in a location and all that. So sometimes we study the plankton samples as well to see whether there are any larva stages in the water. So by studying their density, we can have kind of an idea about if it is a non-indigenous one or either an indigenous one. We can think whether there is kind of a potential threat or if it is an indigenous one, then that's a good sign. So these are some of the things that we need to come up study when it comes to this phyla. So as I told you before, most of the species are sessile. So through the sexual reproduction, they produce these larval stages, and then they completely change their morphology and become an ancestriola. And then this ancestriola will bud and then grow as a colony. So each this zooid will bud and produce different zooid. That is how they grow as a colony. From single zooid to there could be maybe 20,000 to huge amount of this zooid in one colony. The number of zooid in a colony vary, and depending on their age and all that, the number of zooid may vary. Right. So when it comes to their growth, it is important to know a little bit about their reproduction. So this particular species is very important because of their reproduction potential and their different ways of reproduction. So because of this reason, that is why this, even though this particular phyla is unspoken, that is why this phyla is same times very important. So they have fragmentation, that is when the branch of the colony is detached, it will become an individual colony and it will grow. And then this fission, that is colony divided into two. Basically, the creeping one, the one that move a little bit, they do this reproduction through this fission as well. And then the body. That is another interesting thing about these bryozoans. You will will be amazed. They produce kind of something similar to seed, that is statoblast. Once they bud, they will create kind of a layer around it, kind of similar to a bivalve shell. That will become, that can have kind of a dormancy. As you can see here, this statoblast, it can be only seen in Pylactolemata for the moment, and they have this kind of a capsule-like thing, and it will help them to be preserved. So they can, easily, withstand these harsh environment conditions. And even in this archaeological site, we can find these statoblasts along with sometimes pollen collections. So some of these are like sessile one, we call them as sessoblast, and some of these can float, and we call them as flotoblast. So this is another interesting thing about bryozoans reproduction, and interesting fact about bryozoans. And then about their sexual reproduction, they have a male and female in the same colony, and most of the time, first they act as a male and then as female, and then they do the reproduction, and there can be internal or external fertilization. Right. And then when it comes to their colony growth, then there is another interesting about this, their growth. So when it comes to their growth, there is the zooid growth and again the colony growth. So, and again, there is a growth of the colony by extrazooidal calcification. So zooid may grow very in a slow way, and then the colony will grow through budding. And then again, other than that, there can be extrazooidal calcification as well. So that is why when it comes to study about their growth, then again, there's like a huge diversity, and there are a lot of things that we can study about this. So there have one-dimensional growth, as you can see here, and then two-dimensional growth, and in different direction, and then again three-dimensional growth. They may increase the volume. So when we study about their colonies and this particular bryozoan, specifically when it comes to their interesting species of this class Gymnolemata, we can find this different type of growth. So then I believe then you get the question, when do they mature? So specifically, they say that when they have reproductive potential, the zooid with reproductive potential, with the kind of around 30 or between 30 or 200, that then they say that this colony is mature, but this range differs. That could be maybe 2 to like 20,000 or likewise. So when there are reproductive zooid with reproductive potential, the number, it depends. So the mature colony depends. So the next question is their natural lifespan. So you might think other than the, because of, wash away or breaking of this colony, what is their natural lifespan? You might not believe they have kind of a vast range of lifespan. Some of the species that recently came across, I found that they are sometimes 86 years old. So there are kind of a huge diversity when it comes to their age as well. So normally we call the natural lifespan as the larva metamorphosis into ancestula to the at the moment to the moment that when the last zooid dies. The last zooid dies and colonies stop their function. That is what we call as their natural lifespan. So there are like old as 86-year-old colonies. So that's another interesting thing about these bryozoans. So because of their reproductive potential and their ability to grow in any of the available surface, these particular species have become a huge nuisance in terms of economy and ecosystem aspect, as well as in the health aspect as well. So not significant when it comes to health aspects, but economically and ecologically become a huge nuisance. So they may block most of the irrigation line, pipes and cooling system. And whenever we put any kind of equipment under the water, we can find these bryozoans on it. So the best example is this is one of the PVC pipe that I use for my study. I could find them even inside of it. Then you can imagine the situation for this, a pipe system. And again, the other most nuisance thing is the hull fouling. These particular bryozoans, they have a huge capacity to withstand very dangerous chemicals. They can tolerate, these larval stages can tolerate these chemicals, specifically anti-fouling agents that we use for the ship hulls. So what they do is they just settle on it. So they may settle on it and allow other species to settle on top of it. So you can imagine how they are, when how this particular species can become very noisome. Specifically, the larvae of Watercipora and Schizoporella errata, their larval stages can, withstand this kind of anti-fouling pain. And then the other interesting thing about this particular phyla is this, they can attach onto seagrasses and seaweeds and all that. So during our recent visit to Mannar, we encountered some of these bryozoans on seaweeds as well and seagrasses as well. So luckily not this Watercipora subtorquata or Schizoporella errata, but you can imagine the threat that they can pose if any of these introduced species can, grow a lot and become, nuisance. So that is again another very important aspect when we talk about this bryozoan phyla, right? So these bryozoans, and it become increasing concern because of their threat to biodiversity and economy. That is what make it interesting for me as well, other than their taxonomy and their beautiful these zooids and all that. That is what make it interesting for me as well. And the other thing is how we impact for the dispersion of this particular species. None of the animals live on, or the creatures live on this planet is noisome. So it's because of our own action that make it nuisance. So because of us, because of our activities, we create a lot of artificial surfaces. Now the debris become the huge problem when it comes to invasion of this particular species. It creates surfaces. Most of the debris that we encounter during our studies, we found most of the abundance of this particular phyla. So just imagine if in case if it is like a kind of a nuisance species, can you imagine the threat that it can pose through dispersion? So that is why this is like a very important phyla that we need to focus, that we need to discuss. So why these bryozoans, why they are important when it comes to bioinvasions? So as I told you before, they can tolerate to environmental variation. They can tolerate different type of environmental conditions, and they can easily adapt into these different environment conditions. And they can easily settle on artificial substrate. And they have a very lesser larval period, some of them. So they always want to find any kind of a substrate. Whenever they encounter something, what they do is they settle. So what we do is we provide a lot of substrate to them. So if it is a nuisance species, then that's going to be a problem. So, and the other thing is like this less tolerant species. There could be less tolerant species to like, anti-fouling coatings when it comes to this hull fouling issue. So in that case, these bryozoans become helper. So what they do is as they are very tolerant, they settle on that surfaces first, and then it will allow other species to settle on it. So that is why even though these anti-fouling coatings and all that exists, that is why we have to talk about this biofouling or hull fouling problem. So that is why, in terms of different aspects, that is why bryozoans play a major role when it comes to this hull fouling, right? So as I told you before, this bryozoan is not like a simple phyla. It have like a huge impact when it comes to introduce an invasive species. As I told you before, there are more than 100 species that are listed under this global register of introduced and invasive species. And from this, there are 10 species of phylactolematans and 4 species of stenolematans. And from the gymnolemata, we can find 105 species together. So the interesting thing is, I told you this Arbopacula bengalensis or the Electra bengalensis, the one that have kind of rib structures. It is also under this database. It is not, non-indigenous to our countries, but there are other countries that this particular species become introduced. So it is important to know about these aspects as well when we talk about this particular phyla. So here, as I told you before, this species can, grow on any substrate. You can see there are a couple of different species over here. Tube worms are there. And then there is this mollusk species, Ilex tuliper, and then tube encrusting calcareous tube worms. And then on the bottom, you can see Electra bengalensis, but the dominant one in here is this non-indigenous one. You can imagine the threat that they can pose. Not only on my experimental panels, I found them on this bricks as well. I use them as weight. And on top of those PVC parts that I use for my structures, I can't find them as well. And even inside of this pipe. So you can imagine how much they can invade if it is a non-indigenous one. And this particular species can be finding everywhere around the world and become a nuisance. So it can pose a lot of threat to the ecosystem, right? And here is another encounter of this particular species in on a fishing vessel. So you can imagine how much they can pose a threat. So you can see the colony here. And then when it comes to species that I encountered, these were the species that I found as non-indigenous to our country. And Schizoborella aerata was like kind of the significant one that I encountered. So these are a recent finding of we recently published these, and we encountered these in many different debris. Despite of the material and despite of their shape, we can't find them everywhere. So anyway, one of the significant thing that we encountered is they were very dominant in circular shaped surfaces as like a significant finding. So this, we create a lot of surfaces for them to settle down. So we are still on our, we are still working on our species confirmation and identification. So there are like, so far we could find Electra bengalis in these specimens, and there were other unidentified one as well. So we are working on the identification as well. And we talked about their nuisance aspect, right? So they also are very important, like to like the ecosystem as well for other species as well. So they create biogenic structures in areas that are often very flat and sandy. So these branching one, they settle on this kind of areas that are very difficult for other species to live and no hiding places. So they create kind of a biogenic structure so that other species can live, and they shelter and place for animals to live and reproduce. And they also cleanse the surrounding environment. When there's this particular bryozoan, we expect that the water quality is particular, like pretty much good. And it also provide food for large aquatic predators, including fish, sea urchins, sea stars, insect, and snails. Some of the species only eat tiny zooid, and some of the species eat the whole colony. It depends on the species. So here you can see some of the amphipods who rely on these bryozoans. And this is another interesting species that I encountered. This particular goby that were like encountered in Indonesia, they love bryozoan just like me. So they are like, you can see these extended tentacles of these bryozoans. And then I saw a couple of other underwater images, and where this particular bryozoan provides shelter for mollusk, and then some crabs and some shrimps as well. So all of those species were like in kind of a they had like kind of a white coloration that maybe they are camouflaging, right? So other important aspect of this bryozoans is that they create bryozoan reef, just like coral reef. So it acts as a constructor and a binder, and it provides many habitats for other marine animals, as you have already seen. So it will promote marine biodiversity as well, other than if it's not a non-indigenous one. So this reef could be range from few centimeters to many kilometers, and I could found a article from 2022 where there are around 50 uh, biogenic this bryozoan reef in the world. So that's another interesting fact about this bryozoan phyla. So when it comes to this bryozoan, I told you there are many different interesting aspects of this particular species. It's also important as a mineral. So bryozoan minerals, they have they are calcifying animals, and except for Pilectomyllimata, the all freshwater species, and specifically Gymnolimata, and they have a huge diversity when it comes to their calcification. We can find a calcified aragonite, different form of this uh, calcification. So it make it very interesting when it comes to their mineralogy as well. So if you are interested about that aspect, there's something for you to study when it comes to this phyla, right? So the other interesting thing about this is biomol, and it is one of the dominant contributors to temperate water marine carbonate deposit, along with these seashells and all that, and corals and all that. That's another interesting thing about this phyla. And I told you there's something for everyone. So it's again, there are a lot of medicinal importance when it comes to the bryozoan. This is something very interesting when it comes to sessile organisms, because their specific thing is that when they are sessile, they have different kind of adaptations to as a defense mechanism. So for predation, or maybe for kind of a pathogen and all that, they have to stay still. So in that case, they have kind of different mechanism.
Kalpani, we've lost your sound. Kalpani, We've lost your sound.
I'm really sorry, sir. I had a power cut. I just logged in with
Ah,
my phone. Yes, I'm really sorry. Um, just a couple of slides are left to discuss. I will just go through it quickly.
yes, if you
Let me share
can
the
wind
screen.
up, because we've gone over an hour now, you
Yes,
can wind up.
yes, it's
Mm,
almost done, sir.
yeah.
Yes.
Ye-
This is just about their archaeology and paleontology. We can find them in everywhere in archaeological monument and everywhere we can find them. That's an again very interesting thing about these bryozoans, and we can also use them for temperature determination. So, so there are a couple of like a debate regarding their origin, whether they are from Cambrian or Ordovician. This is some of the things that we need to do more research into. And these are a couple of findings that I came across where they are still working on finding whether the origin, but with the Cambrian explosion and all that, we can imagine that they can have kind of a possibility to origin that during that period of time. So as the final thing, I just want to talk about Sri Lankan context. We are little know about this particular species. So there what I use for the comparison is the collection made by Herdman in 1902, and so that is what I use for the comparison. And I used their I had the opportunity to get their publication and compare my findings with them. Or to talk and research about this particular species. That is why specifically that I wanted to discuss during this lecture. So that is just to let the lesser known to know. So that's with that, I would like to line up my presentation. And if there are any questions or clarification, I'm more than willing to discuss those. Thank you.
Thank you very much. Thank you very much, Dr. Marasinghe. I don't see any questions on the chats, but I have a question about the avicularia.
Yes.
Are they chemical protector deterrents, or are they physical? Now, do they have pincers like what the sea urchins
Yes,
have?
they have basically they have kind of a sensing ability as well, but what they're using is that kind of something similar to mandible like that. That is what they flap it, and when there's kind of a predator, what they do is they flap it, and that is what they use as a defense.
Yeah.
And
Yeah.
yes,
Thank you. Well, that was certainly a very detailed and exhaustive thing. I learned quite a lot because I had no idea. And looking back on things, some of the funny things that I have found in the sea would have been bryozoans,
yes.
the foliaceous types, because I never imagined bryozoans were foliaceous. I thought that they were only encrusting on solid
No, there,
surfaces.
yeah, there are different types of morphologies. That is what make it sometimes we
Yeah,
misidentify that is. Sometimes
right.
we can even, yeah, miss most of the fleshy stuff, we can even misidentify this particular species.
Anyone else who wants to ask a question, please unmute and ask.
Yes, Dr. Marasinghe, that with the coral bleaching, do they settle on bleached coral? And
Yes, they can settle on
right,
that.
all right, okay, thank you.
So that would actually slow down recovery of a coral reef. Hello.
Hello, Dr. Marasinghe.
Recovery of a coral reef. Well, a bleached coral is dead.
Yes.
So the coral reef will recover by remnants of the coral polyps that may be hiding in places around. But if it's bleached due to a rise in water temperature, then the chances are that that is dead. And the recovery will depend on new recruitment, fresh larvae settling.
Got it, got it, got it. And what is the difference between diatoms and these things?
Diatom is like this is actually a faunal group. That's the main thing, and that is what most of people misidentify this as a plant. So this is actually a fauna.
Okay.
Zooplankton.
Yeah, zooplankton. Only the larva stage. Yeah, I must say.
I think, Lester, you can wind up now because we've gone
Thank
too
you,
much over
Dr.
time.
Marasinghe. Firstly, I must thank Dr. Malik for hounding her down and asking her to give this talk. It's probably one of the best talks we had in the last couple of years, I think. I see a lot of art. She talked of everything, but there's arts. As an artist, I see a lot of art in these bryozoans, which is very, very interesting. And this is a fantastic talk, and the passion and the information you brought through within this short time is really, really fantastic. And I must thank you so much. And I hope you progress and continue to do this monumental work, Dr. Marasinghe. Thank you so much for your dedicating your precious time to give us a talk. Thank you so much.
Thank you very
Take care.
much. Yeah, thank you very much for inviting me. Yeah, thank you very much.
Thank you.
Thank you, Kalpani.
Thank you, sir. Okay.
Yeah, now you know what I told you last time as well. There are a lot of bryozoans out in the sea, and you have to think in terms of going down and having a look at them yourself
Yeah, of course.
rather than
Yeah,
concentrating on the shipwrecks. Well,
Dr. Malik, Dr. Malik, I remember Dr. Ranjit De Silva was talking about getting old ships and sort of creating artificial coral reefs long time ago around Sri Lanka, and I think he did some monumental work in Southeast Asia someplace. You remember anything about that?
yes, actually, he talked to us about the abandoned oil rigs that he
All right.
saw and worked on in Malaysia. But the Navy has been sinking ships around our coast.
All right. Okay.
Yes,
That's
they've been shifting
that's good. Yeah.
shifted and about and old vehicles. There's one famous shipwreck with lots of the VIP armored vehicles that they didn't want to sell locally.
Right.
They
Okay.
welded them onto the ships so that they won't shift and then
Right.
sank the ship. And that's a popular dive site of Negombo.
All right. I didn't know about that. That's great. Yeah.
Yes.
Fantastic. Yeah. I think you can create a lot of habitat, and it's very interesting. The bryozoans being part of the food chain of
Yes.
fish.
Right.
It's really fantastic. Okay. I'm sorry. I can. Such
Yes.
an interesting
So
Dr.
yes,
Really,
thank
really
you all those who came. We won't be having a public lecture in April, but the next one in May will, I hope, be on seagrasses by Susanta Together.
Yeah. Should be
Thank
very
you very much,
interesting.
everyone, and good night.
Thank you,
Good
Doctor.
night.
Good night, everyone. Thanks for participating on
Thank you,
this.
Harshada,
Thank you very
for
much.
hosting.
Thanks, Harshada. Take care. Good night, everyone.
Good night. Thank you very much.